welcome to the latest Down the Rabbit Hole interview. Today we're going to be talking about extreme environments and the deep sea. I'm joined today by Peter Girguis. Peter, could you please introduce yourself and tell everyone a little about what you do?
Hi, Poppy. Well, first, thank you for the invitation. Again, my name is Peter, or Pete Girguis. I'm a professor at Harvard University in the Department of Organismic and Evolutionary Biology. The research that we do really focuses on understanding how organisms, animals and microbes, have evolved to make a living in the deep sea. And to that end, much of what we do we study these organisms through the lens of genomics and transcriptomics and proteomics. But I'm also a physiologist at heart and began my professional career as an animal comparative physiologist, which means we're really interested in understanding performance. How do animals work? What's their metabolic rate? So too for microbes. What are their metabolic rates? How much energy are they harvesting? How much power do they generate? And how does that shape their fitness, but also change the world around them? So we're a really integrative lab and kind of cross-cut physiology and ecology and evolution.
That's absolutely fascinating. Before we get down to the molecular, let's start really broad. Can you define what counts as an extreme environment? What actually counts as the deep sea?
Well, so Poppy, this is one of my favorite questions because what an extreme environment is really depends on your point of view. So when we as humans talk about extreme environments, what we're really talking about in many cases are environments that are extreme for us. So organisms that live at high temperature, the desert may be an extreme environment, right? Little water or less water and higher temperatures. We view the deep sea as an extreme environment. And in some sense, the deep sea has extremes. There is a lot of nutrient limitation throughout most of the deep sea because they're dependent on the productivity, the plant and algal productivity in the upper ocean that settles down into the deep sea. You know, it's kind of like being at the tail end of a queue in a buffet line. And by the time you get to the buffet, really the stuff that few people want is all that's left. So too in most of the deep sea. So in that sense, it's kind of extreme. Nutrient limitation is an extreme condition. There are other places in the deep sea like hydrothermal vents that in the strict sense of the word really are extreme when you think about our biosphere. They are areas where you have microbes living at the hottest temperatures and acidic fluids and dealing with metals and other toxins. And so they really are at the edge of our biosphere. But to come full circle though, Poppy, you know, when we say extreme, it's really from our point of view. And I would also point out that the deep sea itself is 80% of our planet's living space. So any water that's below a kilometer in depth beyond the reach of sunlight is considered deep sea. And if you look at it through that lens, then arguably they're the normal ones. And we're much more sort of extreme or in the minority from the point of view of deep sea animals. We're the ones who somehow have managed to make a living in this dry, arid environment. So
That's fascinating. And the fact that it only starts a kilometre away, that's, I know, a 10-minute jog. Depends on your running rate. And that's already such a mystery to us. So it's one of our largest habitats on the planet, but we know so little. What are some of the adaptations we are seeing in these marine organisms? You were talking about your interest in the traits earlier, the behavior. Give me the weird and the wonderful.
Oh, yeah, where to start, Poppy? You know, what's weird and wonderful about the deep sea? Well, let's take a step back and talk about what it is. Again, it's that ocean that's a kilometer or deeper. That is beyond the reach of sunlight. One of the most interesting things about the deep sea, though, is that in many places there's plenty of light, but it's light made by other organisms or bioluminescence. Bioluminescence is when organisms make their own light through a chemical reaction, and it's evolved independently, I believe, about 40 times is what bioluminescence experts believe now. That underscores its importance in that environment. And so when I've had the fortune of going on submersible dives, sitting in a tiny little submarine sinking, 2.5, 3 kilometers below the sea surface, as soon as you get down to where the sunlight begins to attenuate, gets a little bit dark, it's just an undescribable light show when you look out the window of the submersible. So bioluminescence is this incredible capacity that is found in so many different phyla. And again, it's evolved independently so many times. That's one of my favorites. I don't study bioluminescence per se. It doesn't matter. I love seeing what people learn when they study how organisms make light. Let me share one fun story about bioluminescence, and then I'll move on to some other really weird and wonderful things. There are fish that live in the deep sea that produce blue-green bioluminescent light. That's the normal color, if you will, of most bioluminescence. And there's a lot of reasons for it. That color, that wavelength, those wavelengths travel well through water, and it's a way of communicating with one another. But there are some species of fish that have evolved the ability to produce red light, and interestingly, to see red light. And we believe they use it like a kind of night vision. So they can shine red light out from these little photophores, these light-producing organs under their eyes, illuminate the area in front of them, and see fish. Most of their prey probably can't see the red light. What an extraordinary adaptation, right? It's like fish have come up with night vision well before humans ever did. And it's this wonderful example of just how natural selection works to produce these capacities that give these organisms that just little edge in terms of their fitness. It's also neat to see how animals and microbes make a living with a little food that they do have. There's that kind of old adage of, one person's junk is another person's treasure. And there are plenty of organisms in the deep sea that make a living off of eating fecal matter and fish scales and just all sorts of amazing adaptations, right? Where they have these sort of low nutrient, low food quality diets, but it almost doesn't matter. Their metabolic rates are low. They're successful. They've been there for, millennia. And they just have this capacity to really hone in on food stuff. Some of them are really specialized. They make big mucus nets to catch fish poo as it rains down. And though that's low quality food, if you get enough of it, you're fine. I often tell students and friends, you can meet your daily caloric requirements by eating a handful of candy bars or a couple of bushels of celery. But if you can meet your caloric and nutritive requirements, if celery is what's around and no one else wants it, there's a solution. So those are two really sort of fun and exciting adaptations in the deep sea. And we haven't even talked about hydrothermal vents yet, which are their own amazing place.
I'm happy to go straight to the hydrothermal vents, and I'm assuming, and perhaps this will take us down to the small stuff. If we're talking about the big stuff, a lot of the bacteria and the microbiome down there as well. Let's talk about the vents and your interdisciplinary work with the molecular biology you're discovering there.
Yeah, thanks, Poppy. So vents are been near and dear to my heart since I was a graduate student, actually since I was an undergraduate. I started off my undergraduate life, just for your listeners here, as pre-med. I really wanted to be an eye surgeon. I really disliked the classes. Also, ironically, I think there's minimum vision requirements for eye surgery, and I don't meet them. Life's funny sometimes. So I bailed on this idea of being a doctor, and really what turned straight to Italian history in the Renaissance. I was very interested in the progress of science, not just in Europe, but around the world. And then I took an oceanography class and was just instantly hooked, especially because of vents. Now, vents were discovered in the late 1970s, around about the time I was 10. And I remember reading about them as a young boy and thinking, oh my goodness, what underwater volcanoes and hot springs, how crazy is that? As an undergraduate, I was really fascinated by them as I was studying marine science. So I went to graduate school to study the hydrothermal vent tube worm Riftia. Now, this is the poster child of hydrothermal vents. Many people have seen them. They're these large, 1 or 2 meter long, big red worms. Let me tell you about some of their adaptations, and then I'm going to move to the microbes, which are really the star of the show. Now, Riftia is this mouthless, gutless worm. It doesn't have a mouth, a digestive tract, or an anus. It is a worm that doesn't eat food in the way we eat food. And this baffled people for a few years. For the first few years, they thought, how does this work? And they thought maybe it absorbs nutrients from the vent water. And they thought it grew so big because it was in this warm, diffuse water around vents. The tube worms don't live at really high temperatures. They like bathtub temperatures like you and I. So what we're comfortable with is what they love. So nobody really figured this out until 2 scientists independently started looking at the so-called guts of the worm, the insides of the worm, and they found bacteria. Now, these bacteria are capable of harnessing energy from chemical reactions. It's kind of what you and I do, right? We take organic matter and oxygen, and we react them, and we harness the energy from that reaction. Only they don't use organic matter. They use hydrogen sulfide. And this is where things get really weird. The bacteria are capable of using hydrogen sulfide and oxygen. So hydrogen sulfide is that rotten egg smell that comes out of sewers. And believe it or not, at high enough concentrations like those found at vents, it's toxic to animals. So this was mystery number 1. How these worms have these bacteria living deep inside them symbiotically that need hydrogen sulfide and oxygen. The worm is an animal like you and me. It uses organic stuff, organic matter, sugars, and you name it, and oxygen, and that's how it makes a living. But how does it get sulfide, this toxic thing, to its symbionts? Well, the worms have evolved hemoglobins that bind to oxygen, like you and me, but also bind sulfide, and they deliver them to the bacteria. The bacteria, in turn, carry out that reaction, harness the energy, and fix carbon dioxide to sugars, just like plants. But they do this in the darkness of the deep sea. And so a lot of my research as a graduate student just focused on those measuring those metabolic rates. How much sulfide is taken up, how much oxygen is used, how much energy is available, and how much carbon is fixed. And so I had the real fortune of working on that and also building and developing one of the first life support systems for these deep sea animals. That's the physiologist in me, studying metabolic rates. But fast forward to my starting this lab at Harvard, I became very interested in the molecular underpinnings. How does this work? And colleagues of mine had discovered evidence that these symbionts fix carbon not just through 1, but 2 different pathways. These are colleagues of mine in Germany who really did some excellent work with proteomics to show that there are proteins that look like they're capable of fixing carbon with 2 pathways. And this immediately caught my attention for 2 reasons. Poppy, believe it or not, this worm with its symbionts are some of the fastest growing animals on the planet. And doing those metabolic rate measurements as a grad student meant that I could measure how fast they grow, and they can fix 10% of their body carbon a day. This is stupefying. Imagine going to bed weighing 100 kilograms and then 110 the next day, and then 121 the day after that. It's insane. And that's what they do. So a lot of our work now is really focused on keeping these animals alive at the conditions they find at vents. But we have built high pressure aquaria, high pressure pumps. We use mass spectrometers to measure their metabolic rates. And then via transcriptomics and proteomics and metabolomics, really look at what are the molecular mechanisms underpinning these extraordinary carbon fixation rates. And that's what our work is right now. And we're beginning to untangle how they use these 2 different carbon fixation systems. When do they turn one up and turn another down, and vice versa? And so we couldn't do that if we didn't have the tools that we have now with transcriptomics and proteomics. But what really excites me is that we can ally metabolic activity and function and the growth of this worm to these molecular processes so that we really have a kind of single cell to animal to community perspective on how they function.
That's absolutely fascinating. And knowing that these techniques are enabling us to see and learn so much more about these almost alien life forms that are so separate from our life on surface is so exciting. A question I've always wondered, how does the circadian rhythm work for creatures deep down, or does it even exist?
That is a very good question. And, Poppy, I don't think there's been a huge amount of work done on circadian rhythms. I can tell you this, though, we do see periodicities in their metabolic rate. And I have an interest, as I mentioned, in how animals shape the environment around them. So we also build a lot of geochemical sensors in my lab, underwater mass spectrometers, that we go to sniff the chemical environment around the worms, a clump of worms, temperature probes that record changes in temperature over time. Now, I brought this up because you asked about circadian rhythms. Well, through our temperature measurements, we were able to see what look like tidal pulses in fluid flow, which means from the point of view of a worm, excuse me, let me try this again, which means from the point of view of a worm that you see these changes in temperature or chemistry. They experience a change that is quasi-tidal. It's a bit offset. This is above my pay grade. I'm not a geophysicist, so I can't explain this. But it does remind us that, the moon is pretty big and actually influences fluid flows and other processes even down a couple kilometers.
That's phenomenal. And to go back into that obviously links with the metabolism as well, but diving back to the ocean microbes as well, what are we learning that might shape our lives above surface as well, and whether that is in our own health or whether this is actually more to do with ecosystems and climate?
Yeah, so let's start with the two worm symbionts, and then I'll share a bit about some free-living microbes that we study, again, through the lens of transcriptomics and proteomics. The two worm symbionts have two carbon fixation pathways, and somehow, together with their host, are fixing carbon at tremendous rates. Now, there's an interest still in thinking about how we might use biological organisms, microbes, plants, what have you, algae, to aid in carbon sequestration. And I think that understanding how these microbes, along with their tube-worm host, sustain these phenomenal rates, really some of the highest ever measured. So they are fixing carbon at rates similar to what we see in the fastest growing algae and plants. And to imagine being able to do that in a bacterial culture, like a big fermenter, offers a lot of possibilities that you might not get with algae. Growing kelp forests to sequester carbon is great. Imagine being able to have a fermenter with microbes that could fix carbon as it comes out of a natural gas plant. So there is this applied bent to the work, and I'll come back to that in a second. We also study free-living microbes that oxidize methane. And there's an awful lot of methane in our world. Most of it is biogenic, meaning it's produced by microbes. And most of it is in the deep sea, and very little of it actually escapes the ocean. But my lab and many others have worked on these microbes that oxidize methane, both with oxygen and without, to ask the question, can we use these organisms to sequester methane, but also to produce a byproduct? If they're harnessing energy from methane oxidation, could they produce a biofuel? Something that is less problematic than just the combustion of the methane. And there's many pros and cons to these ideas. But I wanted to say that in my lab, we really study these microbes that live at vents with an eye towards understanding their fundamental attributes. How do these microbes, for example, lay down the mineral pyrite? We've done research on microbes that deposit pyrite at hydrothermal vents. And get this, pyrite is a semiconductor and can be used as solar cells. not as efficient as the ones today, but if you could figure out how to get microbes to grow solar panels, that is a game changer, because it means you could have, lower efficiency solar panels, but at a fraction of the cost. So the Microbes Advance offer a lot of insights into what microbes can do. And through that, the use again of transcriptomics, proteomics, metabolomics, and I want to emphasize rate measurements, measuring their activity, measuring the geochemical changes around them, understanding the mineralogy. That kind of integrated study gives us a chance to present these observations to colleagues who really focus on the application. And so for me personally, Poppy, I think basic research is fundamentally important, not because and I don't think we should do it with an eye towards application. I think we should do it for its own sake. And here's the catch. As soon as we've completed the work, then we can ask ourselves, how can this apply to what we need to do? And that leads to different observations than going after something with the application in mind, because it tends to bias your perspective. And so I really think basic and applied research fundamentally go hand in hand in that regard.
Definitely so. And it's all about discovery. It feels that this field is really giving us as well. That's so exciting. And you were talking about obviously going down a couple of kilometers and seeing light shows. These environments offer a host of technical challenges as well. So even to just do the discovering requires a huge amount of innovation. What are the main obstacles and the main breakthroughs that are allowing us to explore these deep sea habitats?
Deep sea exploration is not for the faint of heart. There are many ways in which studying Mars is easier than the deep sea. And let me sort of explain, like Mars is a long way away. Even the moon's a long way away. It's about 380,000 kilometers to the moon. But our atmosphere is transparent, or more accurately, semi-transparent to radio waves and to all sorts of electromagnetic frequencies. That's why we have GPS and we have Bluetooth and cellular phones and AM radios and FM radios. I mean, it goes on, but these radio waves and other electromagnetic frequencies can leave our atmosphere and make it all the way to Mars. In fact, that's why we could watch real-time video of a landing of a spacecraft on the surface of Mars. That's bananas. We can't do that in the deep sea. We don't know how. And so, in other words, Bluetooth, GPS, Wi-Fi, none of that works through water. Traditionally, the way we communicate through water is with acoustics, and that means we use modems, just like I used growing up as a kid, these dial-up modems, and they are just as slow, because you know what you're really doing? You're kind of shouting through the ocean, right? That's what you're doing. You have something that is yelling that horrible sound that modems make, and then there's something that hears it. And that's how we move data at very low speeds. Recently, engineers have been designing optical modems for underwater that transmit information through light. And that means you can have something flashing light at a camera, more or less, that is observing the light, and you can move data at pretty close to broadband speeds. Like we can transmit HD video. And we can do that really, like it's getting to the point where we do it really well, over 50, 100, maybe even 200 meters. But I can't shine a light from the bottom of the ocean. I can't even shine a light from 1 kilometer where the deep sea starts. So it's really hard to use remote sensing to study the deep sea. That being said, we're really, as a community, pushing on autonomous vehicles that can be our eyes and ears in the deep ocean. Many of us, my lab included, as I mentioned, we build a lot of sensors and samplers, are leaning on advancements in the commercial sensor space, the little widgets we have for iPhones. They sell all sorts of gizmos that you can hook up to a mobile phone. Or I'm sure you're familiar with Arduinos or Raspberry Pis, these small, low-power computers, these inexpensive open design systems. So we're using those to be able to put sensors in mass in the ocean and leave them there for some time and then go back and recover them. So that is a major advance. So it's not just the highest tech autonomous vehicles that can sail down there. It's also those inexpensive, simpler tools that give us a chance to make measurements and then be able to do it without having to be there ourselves all the time.
That's fascinating. And from the seas to the stars, you've already touched upon Mars and the overlap there. And you've collaborated with NASA. A bit of a sci-fi question, is your work with them about extreme environments and the technological side, or are we preparing to do genomics with astrobiology?
Poppy, there are a lot of scientists out there who are really interested in looking for life on other bodies in our solar system. I'm one of them. In particular, I'm interested in thinking about microbes. We know microbes can survive many of the conditions in space. And we also know that our own solar system has periods where bombardment with, by meteorites is much higher. And you can imagine if life has evolved on Earth, it could be ejected and find its way to Mars. Now, there's a statistical sort of probabilistic framework to that. Let's not talk about that, again, above my pay grade. But as I look at Mars and I look at the ocean worlds, as we as a community think about Mars and the ocean worlds in our outer solar system, they're very compelling areas to look for life. Now, let me tell you briefly about these ocean worlds. The moons of Jupiter and Saturn, there's about a half a dozen or so that have, we think, liquid water. And they are covered with an ice shell. And they may or may not have hydrothermal-like activity. There's a million questions. But the presence of water that's a liquid means that it's in the habitables. That's already a major issue for habitability. What the pH is, the acidity or basicity, we don't know. We don't know how acidic or basic it is. We don't know all the chemicals. But NASA is interested in this as well. Now, here's the thing. NASA builds extraordinary tools for research in space, and even on the moon and Mars and Venus, all of these places. But NASA's mortal enemy, in a way, is water. Water is the one thing they do not want near their instruments at all. They build beautiful small mass spectrometers and other optical spectrometers and all these amazing tools, and they go way out of their way to make sure there's no water vapor because water is sticky. They want nothing to do with it. And a few years ago at a conference, I presented on our own underwater mass spec, and it blew their minds. Like, wow, you can put a mass spec underwater? Of course, I was thinking quite the opposite. Like, my goodness, you made your mass spec the size of a thimble. How did you do that? And we realized that there was an opportunity to collaborate on these missions of life detection. Ocean scientists have tools that NASA could benefit from. And frankly, the converse is true. If we are going to understand ocean worlds in our solar system using the tools that we use to study our own ocean world, makes sense. But more importantly, equally importantly, in my opinion, is that humankind's first life detection missions were actually in the deep sea in the late 19th century. And so scientists have already been through the sort of proverbial ringer in thinking about, is life there or not there? One of the most prominent scientists of the 19th century proposed that the deep sea had no life, the so-called azoic theory. And it was challenged by, frankly, initially non-scientists, right? A very powerful scientist who carried a lot of weight in the field made this assertion, and a lot of people said, oh, well, that's so-and-so, he's really respectable, he must be right. And it took folks outside the field, including a retired medical doctor who was an amateur biologist, to say, wait a minute, that doesn't make sense. And that launched more and more explorations that culminated in the HMS Challenger that sailed from 1872 to 1876 on what is really ocean science's first life detection mission. They set out to look for life through the deep sea. So Poppy, there's this, I think, real natural opportunity for these two communities to benefit from working with one another.
Absolutely. And to follow on in the spirit of collaboration, another passion close to your heart is increasing the accessibility of science. Can you tell us a bit about the work you're doing in this area?
Yeah, Poppy, I think that I'm I really feel very fortunate to be where I am. I'm the child of immigrants from Egypt who settled in California in 1969 with $150. And I grew up in Los Angeles in this aerospace town in the height of the Cold War, where I saw what humanity was capable of. Women and men, people from different walks of life, were capable of extraordinarily things like landing humans on the moon or building a space station or a space shuttle. Now, we did it in large part because of the Cold War. And, in a weird way, I kind of miss that about the Cold War. It brought us together, as horrible as it was. But it sort of baked in me a sense of hope that when we want to do something, we can do quite a lot by leveraging our differences. I don't want all of us to look the same or sound the same or have the same skin color or background. And this is true in science as well. Ocean science or any science benefits from a diversity of perspectives. And I don't just mean, scholars trained at Oxford versus Cambridge or Harvard or Yale. That's nonsense. What I mean is that a scientist from Tonga or Fiji or Bangladesh has a different cultural framework and a different intellectual framework than I do, by definition, and they are equally valid. So to that end, if we really want to better understand this ocean that really runs our planet, we have got to find a better way to promote cross-scientific exchange. And to that end, we do a lot of work in my lab on building tools that are affordable, on providing opportunities to scientists to sail with us on our expeditions, and to try to overcome many of the structural impediments, Poppy, that keep us from working together. There are requirements, for example, on my science funding from the United States that prohibit me from paying the salary of an Indian scientist who's a collaborator. I can't do that, even though their salary is a fraction of what a US scientist might be, or even a graduate student. So there are these structural issues that I'm working on overcoming by working with the United Nations, that's currently developing an international treaty that I hope will promote access and benefit sharing from high seas research. And then also just building tools that enable people to do the kind of deep sea research in their own waters and promote crosstalk with scientists around the world.
That's fascinating. And thank you so much for being a spearhead in that as well. And as we look to the future, what are you looking forward to as we continue to dive deep?
Yeah, I am, I guess, a realistic optimist, maybe, or an optimistic realist. I think that, humanity has a lot of work to do. And I am not so naive as to think that we are suddenly going to become a people that, live, in perfect harmony with the world. Humankind is always going to have an impact on the world. Any living organism is going to have an impact on the world. What I really want us to do is to start thinking about our impact through that lens of sustainability. And I'm really hopeful in seeing earlier career scientists, those that are students and postdocs in my lab, and those that are, earlier in their careers, have a very different outlook than many of us did. And I see a commitment to conservation and sustainability that I never saw, I never felt at their age. I had no idea. And that gives me a lot of hope. If we can get to the point where we treat Earth the way we treat a retirement account, I'll be happy. And let me explain what I mean. When you save for retirement, for those of us who are fortunate enough to save for retirement, you're a banker or planner might say you want to save enough so that you can live off the interest, right? So that this principal, this chunk of money you have, does enough work that it creates enough interest, and if you can live off of that, that would be absolutely ideal. So too with Earth. If we view our planet as our principal, our ultimate principal, and we know that our planet can support so much productivity and create so much food, if we can figure out how to live off that interest without chipping away at the principle, I will be a very happy human. But I have a lot of hope because the scientists of tomorrow are really leaning that way. So fingers crossed for us all. Huh?
Fingers crossed indeed. And I'm going to finish up with a mean question. What is your favorite deep sea critter?
Oh, no, Poppy. All right. So I'm going to qualify that a little bit and say my favorite deep sea critter of today. My
That's
favorite deep
allowed.
sea, thank you. My favorite deep sea critter today um, is probably the see. I can't even do this. I'm actually gonna go back to my old tried and true tube worm friends, and the reason is that not Riftia has many so-called cousins, a worm called Rygia, and there's Lamellabrachia and Escarpia. And these worms live in some really extraordinarily diverse environments. So some live at vents and they live fast and they die young, and they only live for a few years. The cousins of Riftia, so these worms called Lamellabrachia, live at methane seeps. And we think that they live to be several hundred years old. And I love this idea that these two worms that are closely related, and they're symbionts, by the way, are so close, almost identical. And they have such radically different lifestyles. And I think that's really cool because it reminds us in a way, that genomics, transcriptomics, and proteomics are powerful lenses to understand organisms, but they don't tell us the whole story. Because from that point of view, these worms are not indistinguishable, but they're really closely related, but fundamentally totally different lifestyles. That's pretty cool.
That's very cool. So thank you so much for joining me today, Pete. I have learned so much, from bioluminescence to several 100-year-old worms and space as well. And I look forward to eventually seeing the answer to that circadian rhythm question. And I can't wait to see where we all go in the future with this. Thank you so much for joining us and taking us down the rabbit hole.
Thank you, Poppy, for the questions and for the invitation to join you. You have a great day.
And you, thank you so much. Hello everyone. If you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.